Hey guys, Mr. Hyatt here. This is the AP Biology Chapter 5 Lecture Part 1. As you can see on the screen, we're talking about the structure and function of large biological molecules. So these are going to be molecules that are going to be really important in many of the units throughout this whole year. So it's really important that we get a basic understanding, kind of a foundation to build lots of stuff off of. So, what I want you to focus on in each of these classes of molecules are what is it made of, how is it connected, and then give me a couple of examples of each, and those will become clear as we move through these. So macromolecules, macro always means big, so we're talking about large molecules that are formed by joining many subunits together. These are known as polymers because they're poly meaning many, and then this is kind of a root that means subunit. So we're going to connect a bunch of little things together to make one larger molecule. Metaphor for a monomer. So I said a polymer is a macromolecule. A monomer is going to be a building block of a polymer. So this brick wall would be our polymer. The monomer would be each individual brick. Think back to chemistry, and a lot of these reactions are going to be done by condensation or dehydration. So basically, we're forming covalent bonds by removing water from the monomers. Excuse me. The reverse of that is going to be hydrolysis. So we're going to split things using water. That's how we can break our polymers back into monomers. Here's a picture that shows dehydration. So we're removing a water that's causing our unlinked monomer to be connected or added on to the short polymer to make a longer molecule. Then picture 2, we've got our long polymer, we add water, and we break off the end monomer. 4 main types of macromolecules we're going to look at: carbs, lipids, proteins, nucleic acids. You'll get lots more information about each of the 4 of those as we go. Again, know what makes it, what's its monomer, an example or 2, and then what do we use it for. Carbohydrates, remember that A's are things that are definitely tested over, so you want to make sure you know the A-level items. So carbohydrates are used for fuel. You should read that and think energy. Building materials and receptors, and we'll talk lots about those as we move on. Made entirely of carbon, hydrogen, oxygen. The general formula you see there is 1 carbon, 1 oxygen to 2 hydrogens. That again means our carbon-oxygen ratio is 1 to 1 in carbohydrates. So 3 elements make up carbohydrates. Some of these terms are probably familiar. These are special types of carbohydrates: mono, disaccharide, oligosaccharide, and polysaccharide. If you're a strong English student, you probably have a good idea of what these things are going to be just based on these roots here at the beginning, these stems at the beginning of the words. So a monosaccharide is going to be a single sugar, 3 to 7 carbons. Some of them are linear, some of them are rings, but excuse me, I'm sorry. Here are some examples. We've got some ketoses down here and some aldoses up here. They are simple. They're monosaccharides. Like I said, 3 to 7 carbons. There's 3. Here's a longer. Fructose is an example. Glucose, galactose, ribose. Ribonucleic acid. That probably comes up a couple of times throughout the course of this semester. So, depending on where the carbonyl group is, which if you don't remember what carbonyl group is, make sure you go back and watch that lecture. But we're talking about our double bond with our oxygen. If we've got a carbonyl on the end of the molecule, we call that an aldose. If it's in the middle of the molecule, we call it ketose. Most of those I read through. Galactose, glucose, ribose, fructose, those are all monosaccharides. If you notice, they all end in ose. That's a really common word ending for a carbohydrate. So if on the AP exam or maybe on our unit test, I throw a word at you that you've never seen before. If it ends in "ose," you should have a pretty good idea that it's going to be a carbohydrate, so it's going to act like carbohydrates act, basically. Disaccharides, we're going to connect two monosaccharides through a glycosidic linkage, and let me show you what that is on this next slide of the picture. Here's our dehydration. So we remove our water and we get our linkage here. So our water is removed. Notice we're two different examples of two monosaccharides, glucose, glucose. We end up with a maltose, so that's a 1,4 glycosidic linkage. The 1 and the 4 is the carbon that it's connected to. Then if we do glucose and fructose, we end up with sucrose, which is table sugar. We've got a 1,2 glycosidic linkage. Again, it has to do with the number of the carbon away from the oxygen is how we figure it out. That's an organic chemistry kind of thing. You can worry about that when you get to college. But essentially, 1, 2, 3, 4. We're counting our carbons. Over here, because it's a different ring structure, we count in a different way. This becomes 2. So there are some examples, maltose, lactose, and sucrose. You've probably heard of those. Maybe you know someone who's lactose intolerant or something along those lines. That's going to be a glucose and a galactose. Oligosaccharides are still considered they're not huge molecules. They're oligo. The stem means a few. So 2 to 10 simple sugars connected. We see those a lot in our cell membranes. A lot of function in our cells. Polysaccharides are going to be really long ones. Starch, cellulose, glycogen are examples of those. Usually used for storage. Sometimes they help with structure in the case of starch and cellulose in particular. But it's a whole bunch of simple sugars or monosaccharides connected. Here you can see glycogen. That's a pretty. Each of those is a ring, and they're all connected. So that's for sure polysaccharide. So in terms of alpha glucose and beta glucose, think back to our last chapter, the last lecture. I talked about isomers and things along those lines. This kind of follows that same pattern. Notice where the OH group is. Maybe this will help a little better. So in starch, we end up with a layout where we've got a chain where it's up, down, up, down, up, down. Or I guess I should say down, up, down, up, down, up. But notice that the links, the bonds, the shape of the molecule is going to be the same on all of these bonds. Down here in terms of cellulose, we've got an up bond to down bond, an up bond to down bond. So that's going to cause the molecule to be shaped differently, which of course is going to cause it to function differently. Let's talk through a real life example. Starch, which I showed on that last screen, is made of 1,4 linkages of alpha glucose, which causes the molecule to make a helix, and that's where plants are going to store their energy, essentially, is in starch. Here's our alpha glucose, so that's what we saw here. So we've got down, down, down. Cellulose is different. It's got 1,4 linkage of beta glucose, so our OH group is in a different place. So it's going to cause the molecule to be linear, and in plants they're going to use that for structure. Basically, it's part of the cell wall. So there's that picture. Let me scroll up again. There's cellulose. So we've got up, down, up, down. So it's going to be linear. Here's a picture of the beta glucose monomer. The up, down, gets wrapped into a fiber that becomes what we call a microfibril that is going to help build the cell wall. So most organisms can digest starch, but very few can digest cellulose. We eat lots of starches in our diet, but things like cellulose, we can't digest it, so we eat it and then we excrete it in our waste, in one form or another. Yet another example of form and function, you'll see that comes up a whole bunch. Glycogen is related to this, has more 1,6 linkages or branches, really similar to starch. It's essentially our version of starch. You see we find it in muscle and liver cells, provides fuel for us. Remember that's what we said that plants did with starches. That's their fuel, that's their energy source, and that's one of our energy sources. Of course, we know we have to use ATP and all that kind of fun stuff. But moving on from carbs to lipids. Lipids are a really diverse group. There's no general formula like we had in the carbohydrates, the 1 to 2 to 1 ratio. But our ratio is higher in carbon. We've got more carbon than we do oxygen. We're still talking carbon, hydrogen, oxygen, so we haven't worked in any other elements, just those first three. But we've got a lot of carbon and a little bit of oxygen. It's not really a macromolecule like the others because of the fact that the monomer is different and there's no general formula. It's kind of a catch-all kind of thing. There are two types of lipids, fats and oils. Fats are going to be solid at room temperature. Oils are going to be liquid at room temperature. The picture shows a fat that's butter and an oil that's this canola oil. They're made of two kinds of smaller molecules, fatty acids and glycerols. A fatty acid is going to be a long carbon chain. You see 12 to 18 carbons with an acid at the end. And the other end is going to have a CH3, which is very typical of a fat. So we end up with a fat on one end and an acid on the other. You can see here we've got three that are linked, so now we've got a fat molecule, triglycerol. You might have heard your triglycerides level if you've got older family members. I can remember my grandma and grandpa talking about their triglyceride levels when they had blood pressure issues and things along those lines. So 3 fatty acids joined to 1 glycerol makes a triglycerol or triglyceride. It's joined by an ester linkage, which means we've connected an alcohol and this acid group. That's definitely an organic chem thing. You would be expected to know what an ester linkage was in organic chem, probably a little bit above the scope of this course. Something you hear a lot about in the news is saturated versus unsaturated fats, and all that means to a chemist, and all those terms mean are, all these terms mean is, are there any double bonds or not? If something is saturated, then it can't accept any more hydrogen. So it's like if you saturate a sponge, you fill it with water. So if this molecule is saturated, it can't accept any more hydrogens. If it's unsaturated, it can accept more hydrogens because there are double bonds that cause partial negatives and partial positives, all of those kinds of things. So unsaturated fats form or cause kinks in the molecule shape. Let me show you what I'm talking about. If you look here, we've got a saturated fat and fatty acid. It's a linear molecule. We don't have any kinks like we do in the unsaturated. We've got a double bond that causes a kink. Here we have single bonds, so it can be more of a linear molecule shape. So why do fats contain saturated fatty acids and oils contain unsaturated? Well, because the double bond is going to push those molecules apart, which lowers the melting point and impacts the physical properties of the collection of molecules. So this unsaturated fat is going to be pushed apart, and it's going to cause the substance to be a liquid. Where this, we can just stack these up, stack them up, stack them up, stack them up, and make a whole pat of butter. We'll pick up from there in Lecture 2.